Hi, thanks for joining us and welcome. My name is Jack Laser and I'm the co-founder and president of MBF Bioscience, and I'm very happy today to introduce Bruce Kimmel. Bruce has a very impressive career working in academia, pharma, and biotech. He's been the CEO of Vidrio Technologies for 5 years, and he currently serves as the director of the Vidrio division of MBF Bioscience. Bruce combines a deep knowledge of biological systems and integrates that with keen technical expertise. In a moment, I'll share with you a little known fact about Bruce, but first I want to speak about logistics for today. Bruce will give his presentation, and then he'll be very happy to take questions from you at the end. And also, behind the scenes today, we have some magic elves, one of whom is George Jandel, the lead software engineer for ScanImage. So this means you can also ask questions via the Q&A function at the bottom of the screen during Bruce's presentation, and George will do his best to answer them in real time. Okay, so back to the little known fact about Bruce, and that is, for some reason, children seem to really love him. And I've seen this firsthand in stores and parking lots where toddlers and strollers are holding their parents' hands, just smile and wave at him. One theory is that it's because he looks a little like Santa Claus. I think Jerry Garcia. But actually, I think it's because he's a really warm-hearted guy and kids pick up on it. So Bruce is a really smart and a nice guy. What a combination. So Bruce, I invite you to begin.
Well, thank you for that really nice introduction, Jack, and welcome everybody to this webinar. I hope everyone is well and staying healthy out there. And I'm really happy you could attend the webinar, so thanks. We're going to talk today a little bit about ScanImage and how we think we've helped to accelerate neuroscience research over the last few years. And I really want to try to stimulate creativity in you all to think about how you'll use ScanImage to make the next important discovery in neuroscience. So let's talk first about how to, what the talk's going to be like today. First, I'm going to have the first opportunity really to talk to people about the merger with MBF. Then I'm going to focus on multi-photon microscopy and why you should use ScanImage to perform your multi-photon microscopy. And really, ScanImage has the ability to support many different hardware platforms, and we'll touch on that quite a bit. And then we're going to explore some applications that have recently been accomplished with ScanImage in the last few years, and I think that should be interesting for everyone. So first of all, let's talk about the merger with MBF Bioscience. MBF Bioscience acquired Vidrio in 2021, and when Jack and I started talking about this back in 2020, we really felt like there was a strong synergistic relationship between two companies that had serviced the neuroscience community for a long period of time. MBF has 30 plus years in this field. They've been mostly focused in the past on generation and analysis of fixed tissue images. And while Vidrio has been really involved in real-time in vivo brain images and awake animals that are performing behaviors. So we think this is a really powerful combination. It brings more resources to bear, to the neuroscience community. And we hope to bring you in the future new imaging modalities, improve software quality. The 30 plus years of experience that MBF has in writing software could really help improve ScanImage in many different ways. And then, taking that analysis piece that MBF has been working on for fixed tissue and applying it into real-time imaging is another powerful thing we think we could achieve in the future. So I really feel like this is an amazing, combination of two really powerful neuroscience companies. So let's talk about where we are with neuroscience at the moment, right? The cutting edge imaging approaches are allowing us to image brains that are performing complex tasks in real time. And I think a really great example of that is some of the things that Nick Sofroniou did in Carl Svoboda's lab with Microscope, a wide field of view scope. And here, half of the mouse brain in this video is being imaged. The little flashing lights are obviously neurons with GCamp embedded in them. You can segment the neurons and then record from all of those neurons the calcium changes in calcium concentration are happening. And this is really allowing people to monitor networks of 10,000 to 1 million neurons simultaneously across the cortex in alive, awake, behaving animals, trying to correlate that activity then with the behavior that's happening. So that's really where the cutting edge is. So how did we get there? Okay, well, the first types of microscopy, fluorescence microscopy, were wide field of view, and they produce a substantial out-of-plane fluorescence, which causes background and prevents good resolution in some cases. Confocal microscopy can reduce that out-of-plane fluorescence to a certain extent using a little aperture to prevent collection of some of the out-of-plane fluorescence. But it's really two-photon microscopy that came in with a much tighter point spread function that intrinsically eliminates most of the out-of-plane fluorescence. And you've got an example down here of the point spread function of a one-photon microscopy experiment versus two. This really shows you how the point spread function differs, and it's one of the key things about two-photon imaging. So how is that achieved? Well, the two-photon effect is really fascinating. So a typical one-photon excitation, our chromophore fluorescent molecule will absorb a single photon, exciting some electrons in the molecule. As that decays, there will be an emission of a photon of a different wavelength, our fluorescence, which we can then detect. This wavelength of light is typically in the visible spectrum, and only one photon is required to get the excitation. In two-photon excitation, we use a much longer wavelength of light that provides half the energy of the one-photon experiment, meaning that you have to have two photons arrive essentially simultaneously to get the same type of excitation and the same fluorescence. And that will only happen where you have a really high density of photons being delivered to a fluorescent sample. And so on the right here, you can see the true difference between that point spread function, where you've got a one photon experiment here with visible laser light and a two photon experiment here with infrared, an infrared stimulating laser. So that's a dramatic difference. So what does that give us in terms of the properties and advantages of two-photon microscopy? Well, you get this intrinsic optical sectioning, like I was just saying. But because you have this much longer wavelength of light that you're using to stimulate or excite the fluorophores, you also get less scattering in tissues like the brain. So that means you can image a lot deeper in the brain than you could with a one photon type of experiment. Well, that means you need a pretty complicated microscope. You have to have a fast laser, something that's usually firing at 80 megahertz with pulse widths of about 100 femtoseconds, about 10 nanoseconds between each pulse. And then you're going to scan that laser across the sample in a raster pattern using, this type of galvo mirrors. This is what it started with in the old days. And then it's going to hit the sample, fluorescence is going to happen. And then you're going to collect photons, not with a camera, but with a photomultiplier tube. And so what you're doing is recording point-based fluorescence from the sample, which requires you to then reconstruct that image with a computer as you're acquiring the data. So obviously, to be able to do that, you have to have a piece of software that can not only control the microscope, but reconstruct the image on the fly in real time. And ScanImage is just that type of software. Laser scanning microscopy in general can be performed with ScanImage, so you could do confocal microscopy as well. But most people use ScanImage for 2-photon imaging experiments. It's based in MATLAB. It's open source and very user customizable. We'll talk a little bit more about that. You can completely script all the functions that are in the user interface, for example. And it's compatible with most commercial and custom built 2-photon microscopes. So this is not a new piece of software. It's the result of 18 years of development. And the development started in Carl Sabota's lab back at Cold Spring Harbor Labs. Paul Agrutto et al. Published the first paper with ScanImage in 2003. And after that, when Carl moved to the Genelia Research Campus in Ashburn, Virginia, Vijay Iyer moved with him and continued the development for many years, with improving ScanImage 3 and then making ScanImage 4 as well. At that time, Vidrio was spun out of the Genelia Research Campus. It was founded actually in 2013. And, the development continued with ScanImage over the years, so that now we have ScanImage 2021. We have a basic and a premium version that provide a lot of really complex and important features that we'll talk about in more detail now. The software is used all over the world. These are places that that are known to use ScanImage, and one of my favorite is down here, in Tasmania. So I hope one day I can go down there and visit. That seems like it would be a fun place to visit and see how they're using ScanImage. But broadly used across the world, many different countries in Asia, Europe, South America even, and of course the US. So there are a lot of firsts that happened with ScanImage. There were first in vivos that happened with what we would call galvo imaging. So that's mirrors attached to a galvanometer motor and scanning in X and Y, as showed the picture previously. Structural images were taken in rodents initially, and then in 2005, some of the first functional imaging was performed. And carrying on to additional firsts in 2010 and 14, more complex types of behavioral experiments were starting to be performed in real time with 2-photon microscopy and ScanImage. Domdek et al. Using the sort of now classic virtual maze approach with rodents, and then Clancy et al. Starting to do learning types of experiments and really trying to work out, how are these neuronal networks performing in real time. So over the last 5 years, there's been a number of customization projects that we've performed that led to new firsts. One was 3-photon imaging in Chris Shu's lab. Because of the longer wavelength of light, as you can see over here, now you have to have 3 photons arrive at the same time. The light's much longer wavelength and can go deeper in the imaging, and some people are even imaging through the skull at this point without cutting a window in with a cover slip. Also, ScanImage can control spatial light modulators in 2-photon systems, and this has been really important for trying to do simultaneous activation of multiple neurons. In the past, people had been sequentially activating neurons in different patterns with optogenetics to try to interrogate the system. Now with spatial light modulators, you can activate several neurons simultaneously. And then, as we've talked about already, the mesoscope, being able to image very large regions of the brain simultaneously. And something more recent that came out this year that, can be called time demultiplexing. And that's one of the applications we're going to dive into a little bit deeper. But this has set a record. 1 million plus neurons have been imaged essentially simultaneously across a rodent brain. So really exciting developments that have happened. So obviously the software has to do a lot of things. It has to control the scanners. Maybe it's going to control a piezo or a stage to do, the volume imaging. Perhaps there's online analysis going on, maybe even a camera to help align things, etc. So how does ScanImage do that? So there's 3 layers in the software stack for ScanImage. The first is in an FPGA, and the FPGA is essentially the acquisition engine. It's controlling the digitizer. It's taking samples from the digitizer at various rates and then forming pixels based on, which type of scanning is happening, whether it's resonance scanning or galvo scanning. The data is then passed to a C++ layer. This is really important that this layer exists because that's where the data is logged into, say a hard drive or a permanent storage media. And it has to do that with, no loss. It has to be completely lossless, so continuous gap-free acquisition. And that requires some, software engineering to be able to achieve that type of write speed. And lastly, MATLAB is where all the application logic and user interface are. And as we've talked about a little bit, it's fully scriptable. So that's the software stack. And that really allows you to have extensible user functions. You can, program your microscope to do the imaging approach however you want, really, based on the user functions that you add. And you can script every aspect of the user interface into little scripts that you write for further types of automation that you might want to do with your data acquisition platform. And one of the really important things about being able to control the microscope in this way is having a variety of triggers and clocks available to you. This shows that there are different types of frame clock is available, line clock, pixel clock. All these allow you to set up triggers that can happen at certain times. You can also set up triggers with external types of stimuli coming from a behavior apparatus, for example. And the nice thing about ScanImage is that it can be either the director or the worker. So it can be the one that controls everything, or it can be the one that accepts commands from some other external device. So that's really provides a great deal of flexibility for how you control your experiments. Here's a nearly complete list of all the ScanImage features that we currently have. I don't have time, obviously, to go into a discussion of all of them. We will touch base on in a little more detail on the ones highlighted in yellow, but we can do fast Z acquisitions. We can tune your fast Z actuator as well as your scanning systems. We can do 3D motion correction. There's all kinds of features that have come about over the years, and including now some ability to do photon counting in FLIM with the new high-speed vDAQ that's come out recently. So let's talk about hardware and flexibility. ScanImage can support many different configurations. You can get an off-the-shelf system from one of these vendors, for example. You can get kits. There are a couple of groups out there like COSIS and INSS that will help you develop your own microscope if you want from kits or even OEM part distributors. And all of this flexibility allows you to configure the kind of microscope that you want to configure. So whether it's off the shelf or some brand new fancy type of scope that you dream up, ScanImage gives you the ability to really control any of those. So just to dive into that a little bit deeper, here are a variety of different kinds of scanner combinations that people have dreamed up in the past. There's the first one was this Galvo, as we talked about, where you got two mirrors on a galvanometer motor, right? And so you can do nice structural imaging. You can do photostimulation with this type of scanner combination. If you're imaging, you're only going to get in that sort of 2 to 3 hertz frame rate with this type of imaging approach. And, people wanted to go faster, and so resonant scanning became available. So the resonant mirror, as you probably are aware, vibrates at either 8 kilohertz or 12 kilohertz. Maybe there's faster mirrors as well. And this allows you to do much faster area imaging at that sort of video rate, and getting up to 30 frames, 30 hertz, right? And you can start to perform volume imaging that gets you in the 10 hertz range when you start to do some of that. A more powerful combination is to combine the best of both worlds. So now you're combining resonance scanning with 2 galvanometers, and this allows you to do something we call multi-region of interest imaging. You can also do photostimulation, and this gets you into greater than 100 hertz frame rates for imaging, which some people need to be able to do. And I think there's a trend moving forward to go faster and faster with imaging. And that's something we've been observing. And so there are a lot of people that want to try to achieve these higher imaging rates. And then if you get into really, the most fancy systems that are out there, you've got a combination of a RGG, a resonant galvo imaging platform, with a galvo photostimulation platform. And maybe you've combined that with even a metal arc SLM, for example, so you can stimulate multiple neurons simultaneously while you're imaging, see what the impact is, really interrogating the network and how it works. So obviously that kind of hardware has to be controlled by control hardware. And this is the evolution that happened in the sort of control hardware realm. It all started off with this NI6110. Some people still use this. It's only good for galvo imaging, but it is something that still is out there a bit. This was the first card, an Alizar card, that was used for doing resonant imaging. And unfortunately, it wasn't really flexible enough to continue much beyond 2013. And when Vidrio started, we adopted this NI platform, which could allow you to do either resonant imaging or galvo imaging, and had a lot more flexibility. And that was a very good platform for several years. And we tried to work with NI to actually make it even more flexible. There were some limitations still, even with the NI hardware, as flexible as it is. And that led us to ultimately develop our own card called the vDAQ. And there are a lot of reasons we did that. And here's some of them. One of the first things is that we could do a lot better with analog inputs and outputs. So the card comes standard with 12 analog inputs at 1 megahertz and 12 analog outputs at 2 megahertz. And for what we saw is that people kept wanting to add equipment to their microscope, and those are usually analog controlled. And so we had to, have enough of these analog inputs and outputs to be able to help people control whatever complexity they want in their microscope. In addition, we were able to go with a much bigger FPGA in the vDAQ itself, which means that you can do a lot more onboard processing. And that's important, we think, for adding features in the future. We also had a much better transfer rate to the computer. The chassis was limited to about 125 bits per second, whereas the vDAQ can do 4 gigabits per second. So it's got all this same kind of digital I/O as NI hardware would have as well, and some RAM. And this really allowed us to, customize the software around the hardware and provide a lot more flexibility. And one of the things that was really exciting about that is this. We developed a new device widget that's shown over here on the right side of the screen. And here you can see each little device in this microscope has, a little widget for it. And you can tell immediately by looking at it whether the device is working or not. For example, the PMT down here is red. So that tells us immediately we've got something wrong with the PMT and we need to figure out what that is. And this kind of widget coupled with the vDAQ breakout panel allows you to easily configure almost any kind of microscope that you have out there, right? We had one question come in before the webinar started about the ease of being able to configure an existing microscope. And that depends a little bit on the hardware that the microscope has, but these kinds of configuration tools make it really easy to configure even the most complex microscope. So I'm going to transition now into talking about ScanImage applications. And the first one I want to talk about is the multi-region of interest imaging capability. We call that MROE for short. So the real advantage of this is twofold. We call it this sort of postage stamp scanning capability. So if you see the whole field of view of the microscope here, with multi-region of interest imaging, you can target certain areas that you're interested in, and only those areas, and thus speed up the overall imaging rate. And so it's essentially random access imaging within the field of view. It's also very good for effective photostimulation if you're using a galvo-galvo optogenetic approach. And, people have used this type of thing to follow the entire dendritic tree structure of a neuron and watch how signals are integrated at the dendrite and when it results in an action potential being fired, at the cell body. We developed this scan head called the RMR scan head precisely for empowering this, this approach. And it uses the resonant galvo-galvo scan hardware. We did this, we tried to collaborate again with microscope companies and we didn't get much help there. So we decided, okay, we're going to make our own. And so that's available. It's a really powerful device if you're interested in achieving those kinds of imaging rates. So just to go into a little bit more detail, the interface for ScanImage allows you to define the regions of interest that you want. And each region of interest can have a different size, resolution, and power characteristics. There's another question that came in out there, including timing, right? So this allows you to really, look at different areas with different resolution, et cetera. And it allows you to customize it in volume as well. So you can scan one area at one plane in the volume and another area in a different plane of the volume. And so the regions of interest can change size and position with the depth as well. And then ultimately what will happen is you'll image, those areas in the planes that you want to once you've configured your imaging paradigm in the interface. So this is what it would look like for this particular experiment. And you can do more complex, like things that are, shown here as well. This gives you an idea of the flexibility. There are some restrictions, though, for resonant scanning. You cannot change the width and the pixel count for ROIs in a group. And you can't rotate them with the resonant scanner. And that's really a limitation of the physical limitation of the scanner itself that provides those limitations. So this is a really powerful imaging technique that, it can allow you to really get to a much higher imaging rate if you need to. The next thing I want to talk about is an exciting paper that came out on spatiotemporal imaging. So just to introduce this concept with a little less complexity, there was a paper that came out back in 2016 called temporal channel demultiplexing. So first of all, let's imagine that we have an 80 megahertz laser here. So there's about 12 and a half nanoseconds between each laser pulse. And so then let's say we split this with a beam splitter cube into two paths. And one path is delayed compared to the other by 6.25 nanoseconds. So you're basically dividing this 12 and a half nanosecond time frame up into two regions that you can monitor. And so basically then you've got these chain of laser pulses that are split by 6.25 nanoseconds. And so you can point one of them to one area in the sample and the other one to another area. And so you're, in essence, you're with one pulse essentially imaging in two places simultaneously. So this requires, though, that you have to be able to demultiplex the sample. So that means the first 6.25 nanoseconds goes with this, the second 6.25 nanoseconds goes with this area. So you have to demultiplex that. You have to do that in the FPGA. And this is one of the reasons the ultra-scale FPGA and the vDAQ is so important. And you have to sample faster. You have to sample, you can't sample at 125 megahertz. You have to get up into the gigahertz sampling rate. And so there's a fast vDAQ that we made, a high-speed vDAQ that samples at 2.7 gigahertz. So you can divide this 12 and a half nanosecond region into 32 time multiplex channels, theoretically. Okay, so that's the idea. You're splitting the laser beam, delaying one path slightly, and then pointing at two different areas. Okay, well, now let's talk about what Ali Pasha did at Rockefeller University. So they took this to one step more. So they got a slower laser. So they have a 4.7 megahertz laser. But what they're doing is they're splitting that laser into 30 beads. So they designed this complex optical system here so that the light reflects around these mirrors, and there are 30 different delays that are created. So that as you raster scan the laser across the sample, you're essentially imaging in 30 different Z locations along the sample. So this is an amazing thing. They call it the maximum imaging technique. And one other point to make is that as this bounces around in their device here, their delay device, you lose power. So the most powerful pulses are reaching the deepest depths, and the ones that bounce around the most and lose some power are imaging at the surface, closer to the surface where less power is required. All right, so what happened when they did that? Well, they were able to image a 5 by 6 millimeter region down to about 500 microns depth, and with a GCamp6 expressing mouse. And they were able to record from more than 1 million neurons at 2.2 hertz. So I encourage you to read this paper by Tomas et al. That just came out. It's a fascinating display of what what you can do with two-photon microscopy and a customized scan image. Last thing I want to touch on in an application is the activity integration and closed-loop control features that exist in ScanImage. And we've got a real-time video here with imaging happening here in the scan field. You can see neurons flashing green with their calcium indicator. I think this is GCamp6. And this heat map over here is monitoring 50 different neurons across this field of view. And you can see this one just fired, and this one is, here's the action potential that was recorded. So these particular ones here highlighted are ones that are being monitored down here, and you can create a threshold over which if any neuron fires, for example, you can send a trigger to your behavioral apparatus. So that's a powerful thing. What that allows you to do then is connect the vDAQ to your behavioral apparatus, so that if there's a particular neuron that fires and exceeds the threshold, you'll get a trigger to the behavior apparatus. This could stimulate a new type of activity for the rodent to perform. Meanwhile, the I2C packets can be exchanged back to the system to control exactly what changes are happening in the behavioral apparatus, et cetera. And all this can be synchronized at 100 nanosecond synchronization accuracy. And this is really required when you're trying to correlate the activity of these complex neuronal networks with, with behavior that's happening in real time. So this is a really powerful feature. And I think it's one of the most powerful features actually we have in ScanImage at this moment. And I think it's going to really help people dissect behaviors more carefully. So many of these ScanImage features I just talked about are user driven. We've seen this move toward real-time imaging of large neuronal networks and trying to correlate it with behavior, and a need therefore for faster imaging rates. The solutions that exist out there are things like the MROE imaging with an RMR scanner, or what Ali Pasha's lab at Rockefeller has put together, the Spatiotemporal Imaging, requiring a high-speed vDAQ. And real-time activity integration is also really important. The interface allows you to monitor neuronal activity and then send triggers to your behavior apparatus with defined activity thresholds. And I just want to emphasize again that a lot of these features were, driven by users wanting customizations. And, we really want to always hear your feedback about what features, we should add. And, there's been a few questions out there over the last year and a half, and one that came in before the webinar about Python, for example. And one of the things I can tell you is that trying to replace all of ScanImage or rewrite ScanImage in Python is a Herculean effort, right? That's probably not going to happen anytime soon, or maybe even ever. But understanding better how you guys want to use Python, being able to provide, API access to Python or things of that nature would be really helpful for us to understand, so that we can begin to address, your needs that are out there in the user community with Python. So give us your feedback on that in particular. So I talked to you a little bit about how MBF and Vidrio have come together, and we think that'll really make us more powerful in the neuroscience community overall. We think two-photon imaging is the best method for assessing neuronal networks at the moment, and ScanImage can really provide the best and most flexible way of addressing your imaging needs in two-photon. The vDAQ and RMR scanners really bring a revolution into the microscope control and imaging speed realm. So, if you don't know much about those, look at them in more detail. And gosh, tell us about, what custom features will really help you so you can make the next big community breakthrough. I really thank you for your attention, and look forward to any questions you guys might have, or, feedback in the future. Thanks a lot.
Hey, Bruce, a couple questions came in while you were talking, and maybe you can answer a few of them. First one is, can you analyze trigger effects post-acquisition with ScanImage?
So I think there's probably more details we need to understand there, but if you are recording, your triggers, right, then they should be in the metadata of the imaging files, so that so that you can include them in any kind of analysis. But the devil may be in the details there in exactly how it's being performed. But I don't know, Georg, do you want to add anything to that?
Yeah, no, that's pretty accurate, right? So we log all the triggers to the file, and then ScanImage itself does not have a whole lot of post-processing capabilities, because typically our users write their own functions around that. But ScanImage does provide the ability to extract the trigger timestamps from the file so that users can use them in their analysis.
Yeah, I mean, David Tank took that to a whole new level with the I2C stuff, where you can exchange little packets of information with your behavioral apparatus too. So yeah, that's something to keep in mind. It's a good way to exchange small bits of information, in the during the experimental process.
Bruce, here's another question for you. This is from Malia Miller. Can you put those back together in a volume? And I think when that came in, you were talking about the Ali Pasha scanning. I believe that's the case. Malia, if I got that wrong, please let us know.
Well, the answer is yes. In fact, that's where that timed multiplexing analysis comes in. You have to be able to keep track of the little delayed windows of time, right? So in the simple example where I showed the 80 megahertz laser, in the simple example where I showed the 80 megahertz laser being split in two, there you had two 6.25 nanosecond windows. And so you have to, at the software level, divide those. And the signal that comes in the first window goes with the position A, and the second half goes with position B. And likewise with the 30 beads that Ali Pasha's group has done with their fancy technique, each one of those gets binned into another analysis window, right? And all that's done at the FPGA level.
Awesome. And here's another question from Kyle. And I'm just reading this for the benefit of people who are watching this on YouTube video, which, by the way, this recording will be available to watch later on YouTube. But Kyle's wondering if you can provide your slides by any chance.
Well, I'm sure we can. Absolutely.
Great. Well, Bruce, I found that to be a fascinating and extremely informative talk. I want to thank you very much. You have a way of really presenting very complex information in a way that's very understandable. Appreciate it. And Georg, thank you for your time and answering questions as well. And with that, I wish everyone a very, very pleasant day and hope to see you all soon.